Well, good morning. It's Monday, the 10th of February 2020. Now, I've just been reading in the Journal of the American Medical Association, JAMA, a publication they put out on the 5th of February 2020. And I was fascinated because this kind of summarises what we've been looking at over the past nearly 2 weeks now ourselves on these videos. So I want to go through what JAMA is saying, and I want to see where it agrees and disagrees with the conclusions that you and I have reached over these past couple of weeks. There's some new things and there's some things that are confirmed. Some of the thinking, a lot of the thinking over the past few weeks has been somewhat provisional, but it was reassuring to see that this paper confirms it while still disturbing, because it means that things are probably going to get worse, unfortunately. But we need to think clearly about it if we can. Now, JAMA confirms there's 3 zoonotic coronaviruses. Now, actually, this was a bit of a simplification. There's actually 7. There's 7 coronaviruses infect humans. They've all come from animals as far as we know. They're all zoonotic. Zoonotic means they come from animals, and this represents spillover infection from animals into humans. But the first 4 of those are just simple common cold type viruses. They just cause upper respiratory, coryzal, common cold type features. You've probably had them many times. But the other 3 are the severe acute respiratory syndrome, SARS. Now, the good thing about SARS is this was contained, and the SARS virus no longer exists in human beings. It exists in laboratories where people are studying it, which is good. It exists in its animal reservoir, but there's no longer any human cases of SARS. The last one was 2004, apart from one very sporadic outbreak in China, which was quickly dealt with. The other zoonotic coronavirus is the Middle East Respiratory Syndrome, MERS. Now, there are ongoing cases of this, but fortunately the MERS, the Middle East Respiratory Syndrome, is not very infectious. Most cases are direct spread from camels in Arabia, Saudi Arabia largely, and other Middle Eastern countries. So in terms of becoming a pandemic, I'm not worried about that because it's not very transmissible. But the 2019 novel coronavirus, which is this new one, has greater affinity, a greater infectivity, a higher R0. Remember we said R0 is the average amount of, it's the reproductive index, it's the amount of people that one individual is likely to infect. And at the moment we think that coronavirus is between 2 and 3. So for every infected person, 2 or 3 other people can become infected on average as the infection spreads. So the R0 is higher. Now, we originally thought the R0 on SARS was higher than novel coronavirus. It's a bit complicated because it probably is, but the SARS is spread when people are sick in the second week of the infection. So when people are incubating it, they're unlikely to spread. So you don't get what we call this asymptomatic spread with SARS. But with coronavirus, you do, and there's more evidence that you do that I'm going to cover later today, hopefully. Now, novel coronavirus was a single introduction to humans. So the genetics of this shows that this jump from animals to humans probably only occurred once. So one time, a bat gave the coronavirus to an animal, which we're going to think about in a minute, and that animal gave it to a human being, and that's how it started. So it was probably a single infected animal and a single jump of this spillover infection that caused this. And geneticists have deduced this because of the similarity of the coronaviruses that are infecting people. They think it's all the same virus. It comes from the same origin. It's the same clade, as geneticists would say. It's the same group of viruses. Now, the novel coronavirus shares apparently 79.5 of genetic sequences with Sars. So it is similar to SARS in a lot of ways. And in some of our modelling and our anticipation of what's happening in this coronavirus epidemic, we have been using data from the SARS outbreak to try and predict what's going to happen with the novel coronavirus. So, for example, in previous pregnancies, we looked at in previous videos we looked at the adverse effects on pregnancies, which were significant in SARS, causing miscarriage and fetal growth retardation. So, quite an issue, really. So that's still a big concern I have for the novel coronavirus, that this could cause miscarriages in the first trimester of pregnancy, as SARS did. I so hope I'm wrong. 96.2% homology with a bat coronavirus. So it looks like it's come from a bat coronavirus, almost certainly. And interestingly, the novel coronavirus shares the same cell entry receptor, which is ACE2, with SARS coronavirus. Now, what we mean here, I'm not going to go into too much pathological detail, but this virus, the coronavirus, it looks like a crown. So it's kind of got these proteins sticking from its surface like this all the way around. All the way around. Take ages to draw. So it kind of gives it a crown-like appearance on electron microscopy. This is, of course, very microscopic scale. But the point about these surface proteins is they have a particular molecular architecture. So let's imagine that's a blow-up of one of these proteins. So it might look like this. It's got a very specific shape. So that's the top of the coronavirus. That's the top of the coronavirus there. There's the virus down here. And here's this protein sticking out the membrane. And it's got a very specific shaped receptor on the end, or very specific shaped protein on the end, like that. Now, I've drawn a circular one. It's not circular, of course. It's a very complicated molecular shape. Proteins fold into very complex molecular shapes. And this is what immunologists would call an epitope. In simple terms, I would call it an antigen. It is something the body recognises as being foreign. Now, the reason that viruses get into people, what happens is the virus actually gets into. So here's a human cell, which would be huge in comparison. There's a human cell, and the virus has to get into the human cell. So these viral particles, which are probably about this size on an equivalent scale, have to get in. They then take over the cell's genetic material, force the cell to make more and more viral copies. These then bud from the surface of the infected cell, shedding the virus onto the surface where you cough them out and spread them to someone else. So that's kind of what's happening. But to get into the cell, the virus has to get into the cell first. That's a process virologists call adsorption. It has to adsorb into the virus. And the only way it can do that is if this epitope here, this protein on the surface of the virus, actually fits into a receptor on the surface of the cell. So on the surface of these cells, on some cells there's this particular receptor. And as we notice, this particular receptor, not drawn to scale of course, this is a surface protein receptor. We notice that this is circular, so that will fit in. So that epitope there can fit in, like that. It fits in. This is why viruses are usually very species specific, because every species produces different proteins. That's why beef tastes different from pork and chicken tastes different. They all produce different proteins. So this surface protein here is different in every species, but this virus has mutated to fit in human ones, unfortunately. And this receptor here is called the ACE2, and it's found in the lower parts of the lungs. It stands for angiotensin converting enzyme 2. The angiotensin converting enzyme system is a system that's involved in blood pressure regulation in the body, which we won't do in detail. There are other videos if you want to watch that in detail that I've done already in the past on the Campbell Teaching YouTube channel. I've gone into this in great detail, but so this is what happens. This absorbs onto the surface. So this absorbs onto the surface. So if we draw this to scale here, here's the surface protein, which is really tiny. The virus absorbs onto it like this, and that means the virus can get into the cell. And the ACE2 receptors that this virus fits onto are in the lower parts of the respiratory tract. So this actually makes sense now. We've said for a while that the initial symptoms are lower respiratory symptoms in nearly all cases, as opposed to upper respiratory symptoms. It's not presenting as a cold. It's presenting as a lower respiratory tract infection, which can develop onto pneumonia in the lower respiratory tract. But we've also noticed that coryzal features, the runny nose and the cold, often come later on, which makes sense because the virus can sort of spread up from where it originally gets into, and other infections can spread up, causing coryzal symptoms, because the respiratory epithelium, of course, is one continuous surface. It's all one continuous anatomical surface inside the body. But that's how it's originally getting in, through this ACE2 receptor, in the same way that SARS do. That's how it absorbs into the body. There's very specific protein. This is the key, and this is the lock. This is called the lock and the key hypothesis, and this happens a lot in physiology, where there's a particular receptor protein and a particular transmitter protein that binds into it. So this is the key. The viral protein is the key that fits into the ACE receptor lock. That's how it's getting in to the body. So it shares the same cell entry receptor with ACE2, the ACE2 receptor with the SARS coronavirus. Actually, I think there is some difficulty with videos at the moment, so I'm going to try and keep them short. So what I'm going to do is I'm going to upload that one as one video. We'll call that Monday morning, because there's quite a lot of material to absorb there related to the absorption of the virus. And then we'll get back to some more practical things on the next, where we'll finish reviewing this paper and see where it agrees and disagrees with the analysis that we've made so far over the past couple of weeks between you and me, as we've tried to examine the evidence-based literature on this topic.